title, Evan.
Thanks so much. So usually people start off with financial conflicts of interest, and I really don't have any of those, but I would like to disclose the following. So biosafety is a fairly serious subject. And for everyone that knows me, I am not a very serious person. So I would just like to start off by saying, please forgive any of my veiled attempts at humor. But this year specifically, I feel like we all could use a little bit more humor in our lives. So we'll just leave it at that. So because sometimes the end gets a little bit more, so I would say complicated and rushed, I'm going to just go ahead and do my acknowledgments now. We stand on the shoulders, and the end of that is of giants. And so I'd like to start off by just thanking the ISAC Biosafety Committee, specifically Steve Perfetto, Kevin Holmes, Jeff Lyon, and Chris Reifel. Also the ISAC COVID-19 Interest Group, Prateep in Virginia, Chattopadhyay, and also the ISAC SRL Biosafety Author Group, Avril, and specifically Andy Philby, who looped me in on doing this biosafety stuff with that group. And so, you know, kind of like why me? Well, I do fit on all of these committees. I do have 20 years experience. I just did the math. It's 20 years experience working in a biosafety level 2 lab. And so I guess I'm the man for the job. And I also have some experience doing a lot of virology as my predecessor does as well. In the lower right-hand corner, I'm not going to be able to get to everything in this talk. That's the reality. So I leave you with the ISAC Biosafety resource page. So if you click on the tab at the top that says resources, and that will bring you to a number of different things, including the COVID-19 interest group. But the biosafety webpage is there, and that will go over all of the ISAC biosafety standards to try to work to keep yourself safe while working with hazardous samples, and really kind of covers everything. So please do go there as you need it. I'm going to go ahead and stop my video because the rest of the talk here kind of has a lot of, let's say, bandwidth sucking content. But don't worry, there'll be plenty of opportunities to see my handsome face as we go. In fact, I think when I stop my video, my mugshot should show up. But anyway, we'll bring it back up later. So I'd also like to kind of give you my final thoughts at the beginning. And some people may get upset with this, but the reality is, remember, it doesn't matter what I do. It doesn't matter what big fancy University X does. It does not matter what a manufacturer tells you. What matters is what you do with your users in your setting. Nobody can guess what your user is going to do. I'm sure if I did a quick poll here of how many people have had to, say, you know, dig a nozzle out of a biohazard waste bag, I'm sure a lot of you would say, yeah. So you cannot predict what's going to happen. And so the whole point of this biosafety and risk assessment and all of the things that we're doing really is for you to be able to test what's happening in your setting to make sure that you're being safe with your users. And so, you know, let's talk about the 11 million elephant in the room. This is the new abnormal. So this is me back during shutdown. As they say, the flow must go on. And so this is kind of our lives now, right? So with the ongoing pandemic, this is sort of how we're dressing for work. In this case, you know, you got the gloves, you got the mask, you got the waterproof jacket, and that's how it is. But what I like to point out is, you know, really, it's not so bad. And I think the reality is that we're learning to live with it. I'd like to show, you know, this is surgery in the 1920s. And I think when people look at this photo, they'd be shocked to see that no one's wearing gloves. Maybe the one surgeon there is wearing a glove, the one that's actually wearing a mask, but none of the rest of the operating staff are wearing masks or gloves. They're all just in there working on the patient. And, you know, we go from the bad days to the good days. This is an advertisement in the New York Times for Camel cigarettes, suggesting that, you know, more doctors smoke Camels than any other cigarette. It does wonders on your T-zone, and that's T for throat and T for taste, by the way. So I think we all know at this point that these are not what we want to do. And frankly, in the 1980s, HIV really changed the way we think about PPE. I don't think anyone would perform open glove dentistry or anything like that at this point anymore. So as we move forward, we think about new ways to protect ourselves, and certainly in flow cytometry, it's about the same. So what is the 11 million person elephant in the room? Well, it's the virus, SARS-CoV-2. The disease that's caused by it is called COVID-19. Thanks WHO for doing that, just to make it a little more confusing. But HIV and AIDS, I think we get used to it. This map was generated yesterday. So this is your risk of getting COVID-19 if you attend a gathering of 100 people or more. So Tina told me that there would be about 300 attendees, so this is a slight underestimate. But the reality is if we were all together right now and we were in New York City, right there in the middle, we'd have about a 50-50 chance of being exposed to and catching COVID-19, which isn't bad when you consider, where I am, up a little bit further north here in Western Massachusetts / Connecticut, we're basically plague rats. So it's good that we don't have the meetings up there, I suppose. But really, what is new? So many of us have been working in a biosafe environment for a long time, but right now there's two new hazards to think about. And the first hazard is if you happen to be working with virus-infected samples. And so I'll just point out that a number of people have already come up with, sort of the rules and the environments that are required to work with various samples from various tissue types. So BSL-2, with enhanced precautions, is primarily being used when you're working with blood, bone marrow, other tissues. But BSL-3 is absolutely required when you're doing cell culture, growing the virus, when you're working with tissue samples from the lung, bronchial alveolar lavage, nasopharynx, et cetera. But the big deal, and the big deal for us in the flow cytometry and the core facilities is there's this other threat, and that's our virus-infected colleagues. And so how do we protect ourselves against those people, shall we say? And we do that through PPE and physical distancing, and we'll get into some of this as we go along. So how do we protect ourselves? This is not the answer. This is hilarious, of course, but frankly, this just plain isn't going to work. But there are a number of ways that we can protect ourselves, some of which we'll go over. And one of the major things that we do is we perform a risk assessment, right? So we decide how much risk there is to do what we're actually going to do, whether that be return to the lab or whether that be are I going to allow that user to sit right next to me as I, do that dilution, as Vera just mentioned in her talk. And so you got to perform these risk assessments, and who best to help you with that but your biosafety officers and the investigator. And the reality with this triangle, the typical triangle for flow cytometry is, speed, efficiency, and purity. But in this case, it's safety officer, the shared resource laboratory, and the investigator. And there really needs to be open line of communications between all three of these individuals in order to move forward in a safe way. So when you think about the hierarchy of controls, and this is what biosafety officers and internal biosafety committees think about, there's really the most effective way to eliminate hazards is to reduce the risk that you're going to experience is to eliminate the hazard. And the reality for us is that, again, the flow must go on. So we're not going to be able to eliminate the hazard. We're going to go ahead and have to do work either with infected samples or with people that could potentially be infected. There's a way to substitute the hazard, not particularly, no. Engineering controls. Well, yeah, there's a lot of engineering controls that have been administrated, and we'll go over some of those things as we go. Administrative controls. Yep, there's a lot of these things that have been discussed, only allowing one person in the lab at a time, working in shifts, one person per instrument, things that you need to do. And finally, there's PPE. And you can also see on the left there where it talks about going from the most effective elimination of risk to the least effective elimination of risk. But the reality is that the engineered controls, the administrative controls, and the PPE are what we have to work with right now. And so let's just talk about that. So as far as sample handling goes, there's two types of instruments that we work with. There are analyzers and cell sorters. And so on the analyzer, really your best bet is to fix your samples. And fixation will inactivate the virus and prevent it from going ahead and infecting you. on the cell sorter, that's not as easy, right? People don't like to fix their samples and then sort them out and then go try to culture them. It just plain doesn't work. And so the number of engineering controls that have been built into different cell sorters over the years have included these things like, I call it an R2D2 unit, which is the aerosol management option, which involves filtration through ULPA, ultra low particle A. You can also build your instrument into a hood, and that protects you from the sample, right? So that biosafety cabinet is going to recirculate the air and keep anything that is happening with that sample from getting out and getting at you. And then there are a number of other sorters out there. So many of you may be familiar with some of the cartridge-based cell sorters and some of the cell sorters that don't use a droplet. Some of them are shown here on the right. And some of those may indeed be a little bit safer. But again, it comes back to, it matters what you do in your facility with your users. There's always a way to mess something up, and you really need to be able to test whether or not you're actually being safe, and if your SOP is allowing you to be safe while you're handling these samples. So for instance here, this is just a video of a very common cell analyzer. And what happens with this analyzer is that every time you put a sample on, when you're done running that sample, you go ahead and you click this thing called remove tube. And then when you remove the tube, what happens is you put the arm to the side, it comes back and it does this thing called a sit flush, which is fantastic. What it does is it actually cleans the previous sample off completely so that when you put the next sample on, there's no contamination from the previous sample. But the real question here is, would it be appropriate to use an unfixed sample in this situation? I'm going to play the video one more time. So you can see here that that thing is spraying, that thing shot a bunch of droplets towards me. This is me actually recording this video. And so in this case, I think everyone would agree that an aerosol is going to be generated, or at least those droplets are going to come flying at you. And so if those droplets are infected with, somebody's lung tissue or who knows what, we don't really want to be experiencing that in any setting. So when this whole pandemic first started, Isaac, specifically Dr. Costariza and his group in Italy, were the first affected. And they very quickly were able to publish a number of different studies on patient samples, patients that were affected by COVID-19. And this paper that I'm highlighting here talked about handling and processing of blood samples from patients with COVID-19 for safe studies on cell phenotype and cytokine storm. And so in this table here, basically they describe what's appropriate, how to handle blood, how to stain PBMCs, how to acquire those cells on a flow cytometer, and then what to do in the case of unfixed samples. And so here, if you're going back to your IBCs or your biosafety officer at your own institution, this is a good place to start and to say, this is what other labs are doing. So when we do our standard operating protocols, we should consider these different steps. And when we perform our risk assessment, we should say, is this what we're doing? When we train our people, we want to make sure that they're doing their procedures in these environments. Are we doing our staining in a class 2 biosafety cabinet in the appropriate lab? Are people wearing the appropriate PPE, including surgical masks, gloves, eye protection, lab coats, etc.? And so this was an easy way to look at the analyzer. And so I think this has been widely adopted throughout the world. Again, it doesn't matter what anyone else does, it only matters what you do. So you need to be able to test what's going on. And then not long after that, the Isaac Biosafety Committee, specifically Steve Perfetto, Chris Reifel, we were tasked basically with coming up with a standard operating procedure to sort SARS-CoV-2 positive samples. And really the only choice here is to do it in a biosafety level 3 environment. And so in this case, this is again another table from the paper, which is outlined on the right there, procedures for flow cytometry-based cell sorting of unfixed SARS-CoV-2 infected samples. This outlines what's required and what's going on based on the risk assessment. And so, what is what should you be doing to your instrument to make sure that you are being safe while you're operating that cell sorter. And so for SARS-CoV-2, if we're looking at the BSL-3 environment, you need to be doing this aerosol containment testing for each sort. You need to wear the appropriate PPE, and that's the only way that you can ensure you're being as safe as possible. So aerosol containment testing, what is that? Why is that? What's happening? Well, the reality is that all cell sorters create aerosols. I mean, those little droplets that we see in our cameras, each one of those is basically an aerosol. That's the reality. The droplets themselves are between 50 and 300 microns, and then there are smaller satellite droplets. And these depend completely on the nozzle diameter, the pressure, and droplet delays and things like that. And generally, all of these things are captured by the collection tubes and the waste drawer, right? So everything, if we're doing everything right, everything is going to be captured. But the other problem that can happen is, there's these other things called clogs. And again, by show of hands, how many have experienced a clog? Oh yeah, all of you. And so when clogs happen, bad things can occur. That stream will get deflected, and it will no longer be going into that drawer. And so in this video, this is a classic video recorded by Kevin Holmes, where you can see the plume of aerosols being created here in the middle of the video. And this is a waste stream that's just been deflected as a result of a clog synthetically. And you can just see all of the aerosols that are being created. Now, in the absence of the aerosol management option, in the absence of a hood, all of these aerosols are going to be basically coming right at you. They're going to spread from that location, and they're going to be interacting with the operator of the instrument. So it is imperative that our safety controls are actually functioning. So we need that AMO working. If you have a rip in the tube of the AMO, it's not going to work. If your hood, for some reason, they get qualified every year, but at the midway point of the year, do we know that that hood is still working at a maximum efficiency? Who knows? You want to make sure that you're safe. And so to do this, a really simple method was designed by Kevin and Steve to do a quick check of your cell sorter and your safety equipment and your standard operating procedures to make sure that those aerosols are being contained. And it's so easy, and Evan can do it. I mean, that's the reality that it comes down to. The materials that you need a vacuum pump. You have to be able to draw air into something. You need this thing called a rotometer. I did not know what a rotometer was until I was involved with Steve and Kevin, but this is, in short, just a way to measure vacuum pressure. And so you need this to be able to draw 20 liters per minute, which is right there on the old gauge. You need these small beads. In this case, they tested a number of different size bead particles, but they found the best one to use was the dragon green bead at 1 micron. They're relatively small. They can be incorporated into the aerosols that are generated, and they can be collected by the Cyclex D cassette. This Cyclex D cassette is generally used for environmental monitoring for mold spores and other things like that. But in this case, we've co-opted the cassette to go ahead and check our systems to make sure that aerosols are not being generated and being shipped out into the operator's face. You need some microscope slides. Within that Cyclex D cassette, there's a sticky cover slip. That's the best way to describe it, sticky cover slip. And that will, as the air is being drawn over it, that sticky stuff will bind to those aerosols. And if those aerosols contain the dragon green beads, they're going to stick there. You put that on this microscope slide. In this case, we've recommended to use a gridded microscope slide. It just helps for focus for the next thing, which is a fluorescence microscope. And so in the next slide, we're going to go over specifically how to do this in about 2 minutes. Again, it's so easy, and Evan can do it. And this is really how you're just going to make sure that you're being safe, regardless of whether you're working with SARS-CoV-2 or other potential hazards in the cell sorting world. So here's aerosol testing in 5 minutes. Hopefully my audio continues to go through as we do this, but I'm just going to go ahead and try to do this. Special thanks to Jeff Lyon, formerly of Yale University, for his hand modeling in this video. You'll see a little bit from him and a little bit from me. So here we go. Oh, that worked really well. We went right back to the wrong thing here. Here we go. So to begin, we're going to need to simulate a clog. To do that, we stick a split piece of tubing over the waste drawer and go ahead and shut the door. The instrument is on, the stream is running. Once you've placed that little tube over the waste drawer, you're going to go ahead and remove your collection device and go ahead and place a little trough below the sort chamber. We're about to make a giant mess, and that really helps to keep things cleaner after the fact. This is where you're going to sample. You want to sample right here at the opening, right in front of that sort door. We prepare the Cyclex D cassette by taking off the plug at the bottom of it, and go ahead and remove the safety sticker at the top. Now that cassette is ready to go. What we're going to do is attach it to a hose, and I'll just make fun of Jeff here. He's going to go ahead and struggle to get this on his hose. He may have used a little bit of a hard vacuum line here, but either way, it's fine. He gets it on there. Once that's inserted to the vacuum hose, go ahead and turn on your vacuum and make sure that it's going 20 liters per minute, and that's all it is. And then we're going to go ahead and place our Cyclex D cassette again right in front of that sort chamber door. And Jeff's using a little lab arm here to help him with that. I've used a number of things. A brick would work just fine. Go ahead and place those 1 micron dragon green beads on the instrument and load up the sample. Get it running to about 50,000 events per second. I know it's fast. And go ahead and open the waste drawer. That stream is going to hit that tubing that's there, and that's going to simulate basically a clog. And then here you can see the Cyclex D sampling right in front of the sort door, and you can see those aerosols coming out from right below the sort door. Once everything has been sampled, and we'll go over the timing of all that, go ahead and crack open that Cyclex D cassette. And then what we're going to do is remove very carefully that sticky microscope slide cover, cuvette flow, and we're going to stick that on our gridded slide right where the grids are, okay? And then from here, you go ahead and take that and put it in front of your fluorescence microscope, hopefully one that can see green. I think 90% of fluorescence microscopes can see green. And then you're going to count the number of particles that were detected. So what, when, how, where, who do we test, right? So for sorters, you're going to test your cell sorter with all the containment on for 10 minutes, right? And we're going to look at that Cyclex D slide, and you're going to say, how many beads do I have? And what we're expecting here is 0. With all of your aerosol containment on, you should have 0 slides on that 0 beads on that slide. Then you also want to test the positive control, that's fail mode. And so for that, we're going to shut off all of our aerosol containment, and we're going to basically use a new Cyclex D cassette to test that for 2 minutes, all right? And in that 2 minutes, we would expect you to see at least 100 beads on that Cyclex D sticky cover slip, okay? And then finally, I expect you to test your SOP. So how long do you wait before you, after a clog, how long do you wait before you clear that collection chamber, right? So I can tell you what I do, but again, it doesn't matter what I do. I wait for a minute, and that allows that collection chamber to clear out. After that minute is up, I go ahead and I remove my customer samples. Take them out of there, put them in a tube, put them aside, and then I go ahead and I open the sort drawer. And what is that going to do? That's going to actually allow you to clear the sort chamber itself. So remember, there may be a few aerosols lingering up inside the sort chamber that you now need to clear out. And shown here is, the collection device and adapter. There's 3 holes at the back of this thing, and that will allow some of the aerosols to be evacuated from both the tube collection device, and then once you open the sort door, if you don't remove all of this, it'll allow that sort chamber to be cleared out by the aerosol management. There's also been a question of whether or not we should be aerosol containing analyzers. And there is a group of people that are working on that right now. And so where might we want to test those analyzers? And again, we can throw that in the chat and talk about it. But you want to check where the waste is coming into the out of the analyzer, any of the droplet containment. So in the example I showed earlier, once I do that sit flush, there's supposed to be a vacuum that sucks away all of those, that flush saline, and you want to basically check it right there. On other instruments, that's, arm to the side, arm engaged. There's a number of instruments that have onboard mixing, especially high throughput ones. Some of them have vortexes. Others have the ability to titrate or pipette up and down. And all those locations are really where you want to do aerosol testing. And the reality here is that you just want to make sure that you're doing things safely. You're not infecting yourself, you're not infecting your users, et cetera. And all of these things that you're doing, you're basing it on a risk assessment, right? So you have an SOP, you're working with whatever hazard it happens to be, but you need to perform a risk assessment and really go through the entire protocol. And so a lot of the questions that I get is on risk assessments is, okay, how do I perform a risk assessment? What do I do? The first thing I would say is go to your biosafety officer, go to your internal biosafety committee. They know how to do these things. They work in this stuff all the time. That's why your university hired them. But in short of that, basically your risk assessment should contain at the minimum 3 items. One, you want to identify the workforce with the potential for exposure, right? So is that just your staff? Is that your staff and your users? Who is it? Who's coming in and out of your facility? Within that, you want to know what their competency and experience is, right? So it's one thing to say, I have a first-year graduate student coming into my lab and they're working with this brand new virus that they've never done before. That person, is going to have a lot more risk associated with them than, the technician in my lab that's been working in a BSL-2 environment for 15 to 20 years. The next thing you want to do is characterize the risk itself. So you want to include the hazard, identify it, put it in this appropriate risk group. So for SARS-CoV-2, for instance, it's in risk group 2 right now, possibly risk group 3. What is the risk of exposure? What are you actually doing that may expose you to whatever it happens to be? And then also activities that could increase the risk of exposure. And an evaluation and a prioritization. Do we actually need to do this work or not? Is there a way to mitigate this or eliminate what we're doing? How important is it that we do it? Other things, vortexing, putting things on a centrifuge. These are all activities that could increase your risk of exposure. And so you want to identify all that at the get-go. And finally, risk mitigation. What can we do? What can we do to either eliminate the risk or minimize the amount of time that your staff are going to be interacting with a dangerous sample or whatever it happens to be? And within that risk mitigation, you need to have a communication strategy to let everyone know exactly what they're doing. It doesn't help if you come up with all this stuff and you don't tell anybody about it. You want to make sure that you're disseminating that information to the appropriate people. And, this is one of their favorite slides, at least the biosafety people. It's a risk assessment matrix. So basically, how likely is an activity to cause an issue? If it's highly likely and the severity of the outcome is really bad, you go into the red there on the right. If it's something that would almost never happen and also the severity of that happening is really negligible, then there's a low risk in doing that activity. And so this is just 2 ways of kind of looking at it. Is it likely? Is it probable? Is it, occasional, remote, improbable, rare, unlikely? Just different verbiage to really talk about the same thing. And so in performing a risk assessment, and we're going to kind of do that here in a minute together, this is my favorite place to go for risk assessment. It's the Association of Public Health Laboratories. It's my personal favorite. I think it's based on the WHO guidelines, which I'll show in a minute. But they give examples of various states' risk assessment templates, okay? And so to start off, you have Alaska's risk assessment templates. These are based on either questionnaires or fill in the blanks. Again, even an Evan can do this, right? So you can answer the questions, you can fill in the blanks and get the things done appropriately in this risk assessment. There's a Colorado option. There's a Florida one. Yes, Florida has an official biosafety risk assessment. Despite what you read in the newspapers about Florida, man, they do a risk assessment. This is Iowa. There's some stuff, people in Iowa, don't know certain things. Like you can get all your stuff and put it in a bag and like leave Iowa, but nevertheless, if you're still in Iowa, you can do a risk assessment. And of course, New York State Department of Health. This is the most important, I think, to everybody here, that New York State also has a risk assessment template. But again, I think these are all based on the WHO risk assessment. And so in this case, this is specifically from one of their coronavirus PDFs, but it talks about laboratory safety and performing a risk assessment on working with SARS-CoV-2 or SARS-CoV-2 infected samples. And so we're going to go through this one by one as fast as I possibly can, because I realize it's probably not super interesting to talk about, but this will at least give you an example of how to approach a risk assessment and how to bring this to your biosafety officer or to your internal biosafety committee and talk about concerns and what you should do in kind of a lab. And so my tongue-in-cheek approach to this is to perform a risk assessment for working with a SARS-CoV-2 infected user, right? And so, you know, I think the reality is I don't know that we would actually do this. Maybe we would do it with an infected sample, but in this case, we're going to talk about the user. And quick shout out to the SRL COVID-19 working group, because I submitted this to them and they thought this was a great idea to include in that publication, which hopefully will be out soon. So first we're going to we got to do a description of the hazard. So what's the hazard? Agent, SARS-CoV-2. Issue, origin, 2019-2020 pandemic. Susceptible hosts, human. All staff, users, and visitors to the SRL will be susceptible. Disease. Disease cause is COVID-19. Route of transmission, aerosol, direct contact, fecal oral, percutaneous, etc. Infectious dose, I think that's unknown. I certainly haven't seen any publications on it, so that's what we put. Agent stability, it varies. Reports have said up to 3 days, up to 28 days, depending on the surface type and the originating dose. Concentration, again, unknown. What will this work be performed at? Well, we're going to do everything at a physically distant BSL-2. Carrying on, any additional information? Risk assessment assumes that prolonged exposure to infected individuals increases the likelihood of infection. In human populations, infected individuals may shed virus while remaining asymptomatic. Severe risk of illness exists for older adults, people with asthma, or other serious underlying medical condition, especially those that affect the heart and lungs. What procedures are we going to be doing? Well, the procedure here is to safely open a shared resource lab to provide users adequate access to highly specialized equipment and expert service, right? Is this a genetically modified pathogen? No, not in the strict sense of the word. Containment level for all life cycle stages. All life cycle stages of the agent, including prior to disease onset, may result in high virus shedding. PPE, including barriers, gloves, safety glasses, and masks, must be used to prevent unintended exposure. Containment breach. In the event of a breach of containment or exposure to an infected individual, the local environment should be immediately cleared of all personnel for aerosol evacuation, followed by surface decontamination. Personnel should be equipped with a full PPE, as listed above, prior to engaging in decontamination. Pre-treatments and inactivation prior to disposal. Contaminated surfaces, again, must be inactivated with an effective disinfectant for an appropriate length of time. For instance, the EPA-registered disinfectant active against SARS-CoV-2. Lab testing, molecular testing is available, and it's a treat, I can tell you that much. Environmental disinfection for the SRL. EPA-registered disinfectants, which are active and are accepted by instrument manufacturers, shall be used to clean surfaces. Specifically, any high-use areas will be routinely disinfected with, say, ethanol, with a saturated paper towel, and now allowing the area to air dry. Safety controls. So biosafety level practices. Users entering the facility shall participate in the institution's contact tracing and COVID-19 self-monitoring programs. Users will be allowed to operate instrumentation on an individual basis, followed by surface disinfection. All users are required to wear masks and wash hands prior to operating instrumentations. Gloves are highly recommended. Consecutive users, sorry, I didn't mean to pause there, my bad. Gloves are highly recommended. Consecutive users shall be separated by a minimum of 15-minute intervals to allow aerosols to disperse and limit user contact. Engineering controls, including masks, gloves, safety glasses, plastic barriers can be placed in between instruments in close proximity. Clothing, dedicated laboratory clothing and dedicated laboratory shoes are recommended. PPE, all approved face masks and appropriately fitted gloves, clean laboratory coat, and lab safety glasses. Personnel trained on associated hazards. Mandatory training on how to work with the potentially infected users is required for all facility staff prior to commencement of research, and personnel experience. Of course, as we all know, SRL staff are highly trained to work and adapt in an ever-changing world. Finally, in medical surveillance, staff will self-monitor and monitor incoming users and themselves for symptoms. Symptoms can include fever, dry cough, fatigue, shortness of breath, but can also sometimes include headaches, aches and pains, sore throat, nasal congestion, runny nose, etc. Identification of any of these symptoms must be reported, and region monitoring. Researchers from high-risk regions, such as your neighbors to the north here, will be asked to transport samples via courier to be run by SRL staff individually rather than attending the facility in person. So anyone experiencing symptoms should alert the lab director as well as the institutional authority. If infection is confirmed by molecular testing, contact tracing for users up to 3 days prior to an individual's symptoms should occur. Again, each of you can do your own practices. Vaccinations, none are available yet. There's been some great news in the last few days. Post-exposure treatment, contact your medical provider. You have to quarantine for a recommended period. Exposed individuals should continue to monitor for symptoms and quarantine as recommended. And users who develop symptoms must immediately report to the lab director and institutional authority. And so that's just an overview of a risk assessment, something that you would do and share. Share that with all of the people that will be affected by what's happening. And also you kind of put that on file with your institution. But after the risk assessment is done, it's really important that you've documented certain things. So if there's a specific training for what you're doing, you want to go ahead and document that and make sure it's gone, you go over it. These are just two examples from things that I took from the clinical world. This is for doing leukemia and lymphoma immunophenotyping. But on the left is an example of a training document. And realistically here, you want to have this on file. This is what we went over. This is what I trained the user to do. And evaluation has been met in some way, in some form. On the right is our competency. And so every 6 months to a year, you really want to review all of these things and make sure that your user or users or staff are actually doing what you train them to do. And in this case, it's especially critical in BSL-3 and BSL-2 environments that you go ahead and revisit these things. And again, go back and review that risk assessment. Make sure that other eyes get on it, and just make sure that you're not missing anything. And that's something we can discuss even at the end of this, end of this presentation. So other tools for the SRL, of course, masks. I think most of you, if not all of you, hopefully all of you are having your users come in and wear masks while they're in the lab with you for now. Gloves, of course, can be used. I used to have a no glove policy, and I've changed that out. If people want to wear gloves, even to touch the keyboards and everything, that's fine. I'm just going to disinfect it anyway, which brings us to disinfectants. For any of you who are wondering, if you spray the on-off button for an LSR2 with Lysol, the little thing that says, or let me rephrase that. If you spray the low, medium, and high buttons on an LSR2 with Lysol, the low, medium, and high disappear. Just putting it out there. What other tools are available now? Well, you're looking at it, right? So webcams. You can do training by Zoom, WebEx, FaceTime, Google Duo, Microsoft Teams, more and more and more. Cell phones. We're talking to space. It's magical. There are people around the world on this meeting right now. You can troubleshoot from far away. Be surprised how many things you can do without actually having to interact with your users directly. And I think one of these is really important, which is a genuine logbook for contract tracing, should it ever be needed. And the reason that I bring that up is that if you ever have an incident, you really want to make sure that you're not spreading things further down the line. And if you don't know who's been in and out of your facility, there's just no way. There are high-tech versions of this, and there are low-tech versions of this. but you really want to have something available to you. And I'm sure there's more, and let's talk about it at the end of this, that I'm sure we all have stories that we can share. This is just a good resource. This is the list N disinfectants for coronavirus COVID-19. And so if you're using something and you don't know if it is going to work or not work, you know, vinegar and water, but go ahead and enter it here on this website, and they'll let you know if it's been shown to work or if it just plain doesn't work at all. This is a really great resource. And then finally, I'm just going to get back into this. This is my low-tech contact tracing that we're doing at UConn. So basically, everyone that comes into my lab, whether you're signed up to operate an instrument or whether you're just popping in to analyze some samples, or let's just say you're dropping off samples, anything like that. If you enter the door to my laboratory, you are putting your name on that worksheet. You're going to put the time in and time out, and you're going to say, no, I don't have any symptoms. I realize that there's pre-symptomatic people and asymptomatic people, but at least in this case, I want to make sure that we're doing our part to try to do contact tracing if it's ever necessary. And I can't stress enough that the communication between the investigators, the shared resource laboratory, and the safety officer are just critical to make sure that this is all going to work out. I think without that, we're all dead in the water, frankly. So if you don't know your biosafety officer, reach out. They're usually lonely people. They might actually like to hear from you. The investigators may or may not want to hear from you, but that's fine too. And again, I'd like to go back to my final thoughts. It doesn't matter what I do. It doesn't matter what other universities do. It doesn't matter what a manufacturer tells you. It matters what you do with your users in your setting. You just can't tell what some people are capable of. And then finally, I'll just leave you with this slide from the WHO. So stick to the facts, man. Anyway, thank you all for listening. Hopefully I didn't ramble on too much. I know there's a lot of text at one point, and hopefully my videos all came through nicely as well. And I'm happy to take questions as they come.
Thank you so much, Evan. That was absolutely wonderful. That was very informative, and the videos came through just fine. So it was great. We do have a question, a couple of questions that came through, and one of the first questions is asking, how do you modify that Cyclex D testing for a fusion?
Right. So that's the Fax ARIA fusion from BD, which I think I have that right. And so it's really not that different. I would say you have to remove the sort device, right? And is that the one where as soon as you open the door, it defeats the stream? Anyone want to confirm that? You can go ahead and do that.
I haven't been on a fusion myself, so I'll let someone answer that through the Q&A or the chat.
Yeah. I know it can be done for sure. And so if I don't have the right answer right now, we'll get somebody to get to you for sure. But my understanding is once you take away the cell collection device, there's still an opening right there. And you can still set the Cyclex D right in front of that. There's a number of instruments that, you have to figure out a way to defeat the safety. And that's been a challenge for sure. I know some of the Sony sorters, it's basically impossible to defeat those safeties. And so, yeah, those are things that is a problem, and we are working on it. No, but it's definitely a challenge for sure. Yeah. So Tom's saying, yes, the fusion should be able to be done just like the ARIA. So thanks for that. So yeah, once you take the cell collection device away and create that hole there, you can put the Cyclex right in front of it. Okay,
great. And then for that EPA site that you listed that has all the common disinfectants, out of curiosity, does it also list inactivation times and, so how long it should be left in contact with the surfaces?
Yeah, and that's important to bring up. That contact time is kind of critical, and that's what my my biosafety officer is always preaching to me, is that, yeah, you can say you wiped it down with bleach, but then what did you do immediately after is wipe it up with water? No, you got to leave it there for a little while. And yes, the EPA link that I sent you, or that I put on this presentation, has indeed contact times, and there's other, really great information there. So, yeah.
And then follow up to that. So and for any of our SRL members that are joining us today, I think one of the biggest challenges is all the users that come in, right? So you have that tracing or the sign-in sheet and everything like that. That's great. But when we're talking about like enforcement, right? One of the things that comes to mind for me at least is analysis, right? And fixing samples. So you might have some users that are looking for fluorescent proteins that are hesitant to fix. So I'm wondering if you have any ideas or if there's been any thoughts on how we can kind of validate and make sure that samples really are fixed, or if there are any methods in place that we can be confident that we're being safe with unfixed samples on our analyzers.
Right. So, I think the biosafety committee would say, you got to put the analyzer in a biosafety cabinet. I know the answer is not you give it the whiff test to make sure it doesn't smell like formaldehyde. That's definitely not the answer. But yeah, I think short of having some environmental control in there to make sure that you're not interacting with the sample, there really isn't anything. Again, I'll go back to some of the instruments that are difficult to defeat. There are instruments that you can put the samples in a chamber and then have it run. So it's physically separated from you. Now, is there an aerosol management option on those instruments? Maybe, maybe not. But at least you're putting a barrier in there. And then you generate an SOP. So for instance, there's an instrument that you can stick a plate in there and it'll vortex the plate and then pick up your sample for you, right? And so that's fine. You run it, it's away from you. There's a door that's closed. You're not getting in there. But then maybe your procedure is, all right, so once the plate's done with an unfixed set of samples, after all those vortexes have happened, now we need to wait 15 minutes before we pull that plate out and open it up and expose it to us. And how do you do that? Well, you got to test whether that's going to be good or not. And that's where the Cyclex-D, I think, could help again, where basically you test that protocol. You wait the 5 or 15 minutes, whatever it happens to be, then you stick the Cyclex in there and see. And those beads, you can put those beads in anything and collect them. So, we love our beads in flow cytometry, and that's a good set to have.
Yes, we do. And then one last question to that Cyclex-D method. For a novice user, someone that hasn't done it before, how long do you think it takes kind of start to finish, and how often should it realistically be done, for our labs?
Right. And so, I did share, there was a good table there about whether you're in a BSL-2, BSL-2 with enhanced precautions, and BSL-3 setting. And so in a BSL-3 setting, the aerosol testing is recommended to be done at every sort. For BSL-2, it was recommended weekly, or BSL-2 with enhanced precautions. And then BSL-2 alone, that's recommended to be done at least monthly. From start to finish, I'd give it 30 minutes. Once you get really proficient at it, first time you do it, it's going to take like an hour. The longest thing for me, the first time I did it, was finding a fluorescence microscope that an investigator was willing to just, show me how to use and stick it on there. There's a lot of really easy to use microscopes out there, but identifying one and finding one. I know some of us have microscopes in our labs. I'm not one of those lucky people.
All right. Awesome. That's perfect. Well, this was incredibly informative. I think we had one more question from Arthur that came through, but I'll let that be answered through text, and it's just about how many times that cassette can be used. But thank you so much, Evan. That was wonderful, and we appreciate the talk.
Great. Thanks, everyone.
Yes, thank you, Evan. Great talk.